Hi, hello everyone. Welcome to the Anti-Cancer Life, the podcast where we talk about cancer a lot. I'm your host, Taryn Hillen. If you don't know, I was diagnosed with high-grade small cell neuroendocrine cancer back in 2019. I had a slim prognosis rate, less than 10%. So as a former health journalist, I went on a quest to learn everything I could about cancer, and now I help patients navigate their own journeys with resources like this. I am really excited about today's guest. He is approaching cancer and the cancer problem from a very unique angle. He holds a dual PhD in materials science and engineering and nanotechnology, and he's an associate professor in the Department of Bioengineering at Rice University, where he runs a research program aimed at engineering next-generation treatments for a wide range of human diseases, including cancer. I reached out to him recently because his lab, along with several other universities, was the recipient of a $45 million federal grant to fast-track some of their cancer-fighting technology that, according to the press release, could cut cancer deaths by 50%. And importantly for patients, have this tech in clinical trials within 5 years. Please welcome Dr. Omid Veiseh. Hi.
Hello, thank you. Yeah, thanks for having me on the show.
Yeah. Was that an okay intro? There's so much that
Yeah,
you've
that
done
was
in the
awesome.
cancer world. Okay. It's hard to explain to people all the incredible things you're working on and inventing in your lab. When I first read this news about what you're doing with these implants and nano drug factories, I literally thought in my head, like, yes, this is the future of cancer. I think it's so difficult to solve the cancer problem because it's a moving target, it's always changing, and you're actually on the ground inventing things that can tackle it. So I would love to know a little bit about your background and how you got into this work.
Yeah, so I actually started undergraduate work as a studying biology, and so my undergrad degree was in cell biology, and I was really fascinated by medicine, the prospects of coming up with therapeutics. Was thinking about going to medical school, and it became quite apparent to me that doctors are great at what they do, but they have limited tools, and I was really excited about the prospects of building new therapeutics. So I kind of transitioned to engineering because engineers like to make products, and bioengineering is fascinating because it's applying engineering principles to solving big puzzles in medicine and biology. Um, and my lab, as you mentioned, is really interested in developing new therapeutics. And the type of therapeutics we focus on is what we call cell-based therapeutics, and it's a new class of medicines. So when we think about the pharmaceutical and the biotechnology industry today, many of the drugs that are being developed are biologic in nature, meaning that a cell is used to generate them. And this
Mm.
happens in bioreactors across factories, but there's quite a lot of logistics and challenges that goes into actually purifying, isolating those biologic drugs, putting them in vials that then come back to the clinician to administer into the patients. And I've always been fascinated by the prospects of actually bringing that manufacturing into the patient because it opens up so many new possibilities when we think about therapeutics. We can leverage biologics that are really difficult to manufacture and stabilize. We can leverage the cell's ability to sense and respond, which is how biological cells perform. But we have to do this in a way that's safe and protect the cells from the host immune response. So my lab has been pioneering a lot of the work in this area, which is to think about biomanufacturing factories that can be implanted into patients to address these really difficult diseases like cancer.
Yeah, and the thing I loved about it obviously is I went through chemotherapy, and chemotherapy is terrible. It's a systemic treatment. And while it's killing your cancer cells, it's also killing all of your other rapidly dividing cells. And I would stay up at night thinking, how do you solve the cancer problem? Like, I always wanted to have some kind of nano robot that we could train and release into people's bodies that could identify and selectively kill cancer cells and kind of leave our normal cells alone. And so when I started reading about you and what's coming out of your lab, I was like, this is the guy. I was like, he's going to do it. So I kind of want to go a little bit backward and talk about some of your previous research before we get to this new implant device that you guys got the federal grant for. And I kind of want to explain, or have you explain, some of the science of it. So I want to ask you, because you kind of started with drug factories and interleukin 2. So I was wondering if you could kind of break down for people, what is interleukin 2, how we kind of used it as a cancer therapy, but also what were the limitations of using it, because it is very promising.
Yeah, actually, I'm going to start with right where you ended off, which is this prospect of like, what if you had a nanorobot that can go in and identify what the cancer looks like from other cells of the body? And what's fascinating is that the more I learned about how our immune system worked, it became clear that actually our immune cells are very equipped to do this. They are able to recognize cancer cells from healthy cells. And what has been, we're learning more and more, the field has now developed immunotherapy drugs, which are transformative, much different than chemotherapy. Chemotherapy is designed to kill fast-growing cancer, and as a byproduct, unfortunately, kills a lot of healthy cells. But immunotherapy boosts your immune system to do what it's supposed to do anyway. And part of the reason why cancer developed is actually a failure of the immune system. So we're working on coming up with molecules, these are drugs, that activate the immune system in a manner that would effectively prime them to recognize and eliminate the cancer. So as you mentioned, some of my prior work, again, when I started my lab at Rice University, we had a family friend who was diagnosed with ovarian cancer. And it's one of those really difficult cancers where the prognosis is not great. And I was right across the street from MD Anderson, and I ended up connecting with a gynecological oncologist, largely to learn more about, what are the options for this disease and, what next generation therapeutics could look like. And he pointed me to a trial. So I met with Amir Jazairi, who is actually one of the investigators of this Thor project, which we're talking about today. And he runs a lot of clinical trials and is fantastic clinician that manages patients with ovarian cancer. And learning more about the disease, and what he kind of explained to me was, it is a failure of the immune system. And there's evidence in the literature based on work that had been done that if you did activate the immune system properly, you could actually, eliminate these type of cancers. But the challenge is how to do that in a safe and effective way. And one of the motivations for our work was this work of Edwards at University of Pittsburgh, which happened decades ago. And there's a molecule that the body itself produces, interleukin 2. This molecule is a very potent activator of a specialized type of immune cells called T cells. Now, T cells are like those robots that you described. They have the ability to recognize friend from foe, and in this case, the foe is the cancer cells. But they need IL-2. IL-2 is the molecule that activates them, that allows them to divide, that allows them to get properly trained to what the cancer looks like. The challenge with IL-2, which is a drug that's available today, again, it's a molecule that the body itself produces. So it's been several decades now that it is available in the form of a drug called Proleukin. And Proleukin works, it's just incredibly toxic. It works in certain kinds of cancers, but it has to be administered in an ICU, and it has a really short half-life. And when we think about drug delivery, what we're doing is we're usually administering them intravascularly, meaning they go into your bloodstream. And in your blood, you have large reservoirs of lots of immune cells. So if you activate them all at once, that's not ideal. It causes a lot of the toxicity. And so patients have to receive this in an ICU. They get these drugs administered every 8 hours. And it's really a miserable experience, unfortunately, because it makes you feel like you have like the worst flu of your life. And many of the patients, unfortunately, are not able to complete the course of treatment that's needed. And so they are not getting the benefits from a molecule like IL-2. But what had been done by Edwards was he realized that if you administered these molecules locally, that's how the immune system uses them. They're produced locally, and it activates the immune system locally. But getting drugs to where the cancer is getting this molecule to where the cancer is in the abdomen is actually difficult because it has such a short half-life. So we thought this is an opportunity for us to leverage our biological drug factories, and we came up with IL-2 producing factories that could be administered to the abdomen, and they reside there and produce interleukin 2 for a programmable amount of time. And by producing them locally, we keep them away from the blood, which causes the toxicity. And so this is a project that we started in my lab in 2018, and we were able to get this into human clinical trials by 2022. So now we have this trial active at 5 different sites across the country. We started this company, Avenge Bio, that's sponsoring the trial and running it right now. So we're hopeful about that approach, but it also became apparent to me that maybe that's not enough because we need to have...
Yeah.
Yeah.
So are you running that trial with the interleukin 2 drug factories? Is that on ovarian cancer as well?
Yeah, it's specifically on ovarian cancer, and it's for patients that have already received multiple prior lines, including chemotherapy, and it hasn't worked for them. And so it's a difficult stage of their disease. We're really treating people that unfortunately don't have a lot of time left, and the hope is that perhaps we can really alter the course of their outcomes through this approach.
And this is important too because, and please correct me if I get this wrong, but the peritoneal cavity is difficult to get into, right? To sometimes get those drugs into that area, which is where a lot of your organs and including your ovaries would be.
Yeah, many cancers, in particular ovarian cancer, metastasizes to the peritoneal cavity, which is really kind of, you think about it, your abdomen where all your organs are. And just the nature of that cavity is that, drugs don't naturally seep into there. Immune cells that are there tend to be of the suppressive variety, meaning like, we have the immune system as a balance of inflammatory cells that can eliminate cancer, but suppressive cells that can, suppress inflammation. And so it's an environment which unfortunately is very hospitable for cancers to grow because of the suppressive nature of it. And so if we can change the immune dynamics and properly train the immune system to recognize the cancer there, I think the results could be quite transformative.
And how long does a clinical trial like that take? Is it because you have to follow people, obviously, for, a 1-year check-in, maybe a 24-month check-in, and especially if it's stage 4 patients, you might be looking for, just longer survival times. When would you expect to have data on that?
Yeah, so that trial is ongoing now, and typically for a trial like this, you'd want to follow patients for 3 to 5 years and see how it's having it. The first priority is to find the right dose, which is actually one of the things that we realized is the goal of our first trial, is to figure out what is a dose that's maximally safe for all the patients. And for the people in the audience that may not be familiar, many clinical trials get done in what's called a dose escalation manner, which means you might have we can simplify this and say, at each dose interval, you start with the lowest because safety is always the highest priority. You treat 4 patients, and if none of them have a response, meaning a detrimental safety kind of signal, then we would graduate to dose level 2. And you do this until you get to what's called the maximum tolerated dose. Now, with immunotherapy, this is really tricky because there's a really fine window, it's a very narrow range between efficacy and toxicity. And one of the things that we're realizing is tough with immunotherapy is when you do this trial and say one patient has a response, because of the heterogeneous nature of the diversity of the human population and immune cell responses, it might be tough for that one patient, but actually for the other three patients, it might be insufficient. And so that was the inspiration for Thor, is realizing that, okay, like dosing is really complicated with immunotherapies, because what might be an effective dose for one person may be totally off for the next person. And we don't really know this in a clinical setting. We really need to have that feedback regulation. And so that was why we came up with this idea to think about the next generation for Thor. But right now, the trial that's active is going through that dose expansion phase. So far, it looks to be safe, but we're kind of pushing on the dosing until we get to what is the maximum level that is.
Well, thank you for explaining that, because I think patients are largely in the dark about how clinical trials usually work, especially in next gen technology like this, of what you guys are actually looking for from phase 1 to phase 2 to phase 3. So I think people will really appreciate hearing that.
Yeah.
And it also leads us into your next invention for what you guys got the grant for, this Hammer and Thor program. So just to recap for the audience, because I know a lot of science is being thrown at them, I think people understand that immunotherapy, it activates your immune system to help fight your cancer. But as anyone with autoimmune disease knows, you don't necessarily want an overactive immune system. So when you're giving something like interleukin 2, it can almost overactivate the immune system and cause toxicity. And it's also difficult to give it systemically because, like you said, you have to be in the ICU. With your drug factories, do patients still have to be in the ICU, or are they actually able to go home because it continues to give them interleukin 2 locally?
Yeah, so it's been great. I think the patients have largely felt it was really well tolerated. They get the treatment and they go home the next day. And it's a living drug factory, so the drug is produced locally over the course of, in this case, it's intended to work for 2 to 4 weeks, and then they come back for scans. And yeah, it's sort of uniquely different. And I think some patients, they're used to you go in frequently for your chemo and they're like, I don't get this. I just get this and then I get to go. And it's yeah, because even though you're not here, you're still getting the drug. It's just being produced inside you, which is kind of a different cost.
And that's so amazing, because I had 6 rounds of chemo. I had 21-day cycles, and I did a round where I was stuck in the hospital for 3 or 4 days. And it's awful. When you actually have to get chemo in the hospital, you're getting it at weird times. You're just not as comfortable. Your family's not with you. And I do think these small things of you have now allowed people to go home with the drug in their body. That in itself is a huge quality of life, win on its own.
Right.
So let's talk about the next iteration of this technology, the Hammer and Thor, which I love these names that you guys came up with. This is fantastic. It's great marketing for this really amazing technology and science. And so I hope the whole world gets to learn about it. Can you explain we'll kind of do the broad stroke overview of what this device is, and then we can kind of get in the weeds on it.
Yeah, the inspiration for the work really came through my own observations, and as well as many of my colleagues who are running these trials. And it's you dose the patient, everyone's anxious to know if it's working, and cancer today is managed as if it's a static disease, which it's not. You can relate to this, right? I mean, you go on a course of therapy, and you keep getting the drug, and then there's some kind of imaging that's done weeks to months later, and then there's sort of an understanding of well, maybe it works, maybe it doesn't. And if it doesn't work, you can't get that time back. And so one of the things that was really, puzzling, that makes this really hard, is you need to have a way to track in real time what's going on within the tumor, and talking to the clinicians, all the stakeholders. I mean, the patients want to know. The developers, we're anxious to know. And the clinicians, they're well, I don't know if, what we're doing is right or wrong until we see your next scan. And then by then it's kind of late, because tumors grow really fast. And now it's a much more difficult disease to manage. So this was the inspiration. We were like, if we could come up, and the truth is, like, there's a lot of technology innovation that's happened, but what really was missing was integrating these pieces together into a solution like Hammer, which is an implant that has on board, it's a hybrid bioelectronic device, meaning it has a bioelectronic component as well as a living cell component. And these two pieces work together. The sensors that are the electronic component allow us to look for biomarkers of safety as well as efficacy. And then the cells produce the immunotherapies. And by having this more closed loop, we can just implant the device. The device communicates through wireless technology with the clinician, and it tells them, is this the right dose for this patient? Is this the right immunotherapy for this patient in real time? So rather than making changes, weeks to months later, the very next day, you can change course because you're like, this is not the right dose. They're not seeing, the response they should be. We need to up this. And all of this could be managed at their home because they just get the implant and then they're free to go, which I think really enhances access to clinical trials. Right now, if you live in a major city like Houston or New York or San Francisco, you have, really top medical centers where lots of trials are being done and you can access them. But if you don't live in one of those cities, it's a big burden to kind of be attached to a city which is not your hometown. It's a really difficult time in your life where you really need to be close to family and as much normalcy as possible. So having all of this remotely tracked and managed, I think is quite different than how medicine is done today.
And do you think it helps solve the problem of the individual response to immunotherapy of would you guys be able to see in real time, like at this dose, this patient's responding great, but at this dose, this patient isn't. So we're going to up the dose on this person rather than having to have these static doses of like, okay, in round one, we're doing this and then we're going to escalate up. Does it help you? It becomes a more dynamic process.
Exactly. That's exactly how it is. And it actually has, initially it's going to be all clinician guided, meaning the clinician will make those decisions. Mm-hmm. But we also have already working with a group at Georgia Tech that's incorporating artificial intelligence. So just like, your smartphone is now able to predict what, the next sentence should be based on what you're writing, it's going to learn. And as you treat patients, from the first patient till the last patient, you collect so much data and then you start to notice patterns that, maybe this is actually the way to go is cycles of maybe a high dose and a low dose as opposed to like, what is the maximum dose that they can tolerate? And so that's going to be the exciting thing is just as you do more and more, it's going to get better and better in terms of how robust the technology is.
That is the part that makes me so excited because I know a lot of times we're using genomic testing now. I had Foundation One genomic testing on my tumor and you get that big sheet back that basically tells you, here's all the mutations that you have that are individual to your cancer. But again, it feels very static because it's like they took that biopsy at the early stage of my cancer. Right. We don't know what micro disease was mutating, how it was changing as time was going on. And also, if you look at the printout, most of it says, we know this is a KRAS mutation or this is an EGFR. We don't have clinical relevance yet. And right. I just like the idea of all the data you guys could collect. I can't even imagine in a world where if every cancer patient had this implant and we could just in real time be figuring out, hey, everyone responds really well who has these mutations. Like look at, kind of how they developed Herceptin back in the day when someone like put it together that, people with HER2 receptors had worse prognosis rates for breast cancer. Like that takes so long to, 20 years to develop those drugs and those antibodies that can fight that. And now you guys are coming along with something that I think, if all goes well, you could fast track that data collection and we could be seeing things in real time, which is very cool.
Yeah.
I wanted to ask you No,
I was going to say, I think that's exactly the point because a lot of investment in biotech, let's take obesity for an example. Right now we have all these blockbuster drugs that are treating obesity. Well, these are not new molecules. In fact, they've been around for decades. But what's different is the pharma companies were forced to invest a lot of money into optimizing the logistics of how do I, because the natural molecules that the body produce only have about a 2-minute half-life. So they were never really druggable because you'd have to constantly inject them. So the drug delivery piece is the huge bottleneck and a big source of like investment by pharma. A lot of their money is going there. But actually, you're right. I mean, what we need to understand more is more targets, more therapeutics, a platform where, if we, by creating these biological factories, we've sort of eliminated the need for all those logistics of how do we create an injectable that lasts for like 2 weeks or 3 weeks. Here we have the living cells, the biofactories inside the patient. It can make different drugs. It can make different doses of drugs. Now it opens up so many new possibilities that allows us to really do the iterative testing, like you're saying, learn from the heterogeneity of the patients as to which likelihood, which particular, perhaps mutations or just different aspects of their immune system could predict better efficacy if they got a particular course of therapy.
Yeah, because I think, one issue that always haunts me is that immunotherapy is so promising, but when you look at the stats on it, like I barely qualified for immunotherapy, so we didn't do it. My PD-L1 markers they were like borderline. So it was like, maybe we could give you a checkpoint inhibitor, but like the odds of it working are so low. And I know sometimes people, it's like, that's their next line because that's all that's left to do. And that's so frustrating to put people through treatment where they're having to make decisions like, well, this has a 5% chance of working. But we're going to, you're going to pay all this money and it's going to be all this time and it's going to be exhaustive. And I think as we're getting more data on immunotherapy, it's like only 50% of patients qualify for it right now. And then of the patients that do, it only works. People only have like a real response in something like under 25%. So right now it's this very promising therapy with very low patient population, patients being able to qualify for it. So I feel like this just opens the doors to figuring out who qualifies, who we put it in, and how we change it to make it more effective.
Yeah, exactly. I mean, unfortunately, you're correct in that, in 50% of tumors have sufficient mutations, which would predict that the likelihood is that the immune system would be able to recognize these cancers as foreign and be able to eliminate them. But then immunotherapy is necessary to prime the immune system properly. And right now, with the current immunotherapies, only about like 5 to maybe 15% of solid tumor patients respond to it. And so that's the motivation for this project, is how do we broaden that so that it works in all 50% of the patients that it's supposed to work in. And what's unique about what we're doing versus just, statically administering the maximum tolerated dose, which was likely the maximum tolerated dose for a subset of the patients in the trial, right? There were probably some that saw toxicity and it worked for them, and that becomes a dose for everyone now. But actually, the immune system is so heterogeneous that we do need to think about each individual's, and even that particular individual at that moment could have a much different immune response with a different dose of the drug. People have probably experience when you get a cold or the flu. Sometimes you have a miserable response. Sometimes you have, a more muted response. And a lot of that has to do with like how active and primed your immune system is in that moment of time.
Yeah, and the device you guys are working on, and is the device Hammer or is the device Thor?
The device is Hammer, and the project is...
The device is Hammer, okay.
Yeah.
The project is Thor. I just wanted to get that right. Okay, so in Hammer, you guys you're basically also measuring around the tumor microenvironment, correct? Is that right? That it's going to be able to send signals back about the tumor microenvironment?
Yeah.
Can you explain to people why that's important? Like how the tumor microenvironment kind of puts, it helps cancer win in a way because it's constantly changing and cancer is always using it for its own benefit, not yours.
Right. Yeah. So, again, with immunotherapy, if the immune system does its job, people have, it's just part of biology where we always have mutated cells that just grow in the body. But when the immune system is working properly, it's able to find those and eliminate them before they grow and become a cancer. So cancer is in a sense a failure of the immune system. And the way that the immune system works is you have lymph nodes where immune cells pick up kind of foreign antigens, go and become activated and primed. It's where they learn what antigens they should be fighting. Now, cancer cells that grow, they have certain mutations which allow them to kind of evade the immune system by producing cues that kind of suppress the immune system. And the only way to fight back is to produce the counter molecules in those adjacent lymph nodes, so you can actually properly now educate the immune system to go and eliminate this cancer. So that's why this is such an important part of the puzzle, is finding what aspects of this tumor is causing suppression and countering those with the right immunotherapy for that patient in that moment in time.
I don't know if this is correct. I always think of it as like cancer cells hiding in plain sight, like cloaking themselves and saying, nothing to see here. Like, I'm totally a normal cell. And then our immune cells kind of pass by and are like, oh, that's a normal cell. Don't,
Yeah.
don't
it
kill
is a
that
bit
one.
like that. It's exactly that way, because the immune system is also always worried about attacking itself, right? So that the other end of
Right.
the pendulum, which is the autoimmune aspect. So there are mechanisms by which our healthy cells produce signals that tell the immune system not to attack them. And unfortunately, cancer cells hijack those same signals. So we just need to kind of renormalize the immune system. And by doing so, we can effectively treat cancer. Like, that there is real world evidence in the subset of patients where immunotherapy works. It's a durable response,
Yeah.
and the tumor doesn't come back. And it's really amazing because, with chemotherapy, you always have kind of mutated cells that come back. But the beauty of immunotherapy is once you educate the immune system as to what the cancer looks like and you properly prime it, then it will not only recognize those mutations that exist at that time, but it has the ability to recognize newly emerging mutations and eliminate the cancer altogether. Even if it comes back later, it's now primed and knows what it looks like to kind of eliminate it.
And that's so incredible because, for me, high grade small cell neuroendocrine, the whole danger of it is it's not getting to NED. You can get to NED with like a surgery or sometimes, with cisplatin or platinum based drugs. The problem is recurrence, and it has a 90% recurrence rate for the type I had in the uterine cervix in the first year. It's like they expect that it's going to come back. And I think that's what makes high-grade small cell neuroendocrine like a very tricky disease, because we have this weight of recurrence just hanging over our heads. And we know that the chemo did not teach our bodies how to fight those cancer cells. So this idea of being able to fight more things with immunotherapy, if we have real-world knowledge about, what it works in and getting that data in real time, is just so incredible. I wanted to ask you if the next iteration, if at some point with this device, if the next iteration, would it be measuring, additional cancer biomarkers, not just sort of like the response that's happening in the tumor microenvironment in relation to, upping the dose or lowering the dose, but would you be able to see in real time, like, oh, there are even more, X receptors on the cell now, or like there's even more of this protein being produced and there's even more of this, these elevated numbers are now telling us that there's more of this thing happening. Like, is that sort of on the docket?
Yeah, I mean, I think like that's basically why I think ARPA-H was excited about investing in building the platform, because once you build the toolkit that allows you to monitor things in vivo and produce drugs in vivo, all of these elements are plug and play. You can easily replace them. There are certain cancers that have biomarkers, like, with ovarian cancer, once people have had surgery and they remove their ovaries, CA-125 is a good biomarker because that's a particular hormone that is produced by the cells of the ovary, and people that have had their ovaries removed, baseline should be like almost zero. But if the cancer comes back and you start seeing measurable levels in the blood, that tells you the cancer is growing, right? So there's basically those biomarkers that are important to track. But yeah, also specific subtypes of maybe mutated cells that are coming back. I think that's the power of the platform, is if we can do these things, and not just even for cancer. We can do the inverse with autoimmune diseases for people that have
Right.
lupus, inflammatory bowel diseases. We can swap in the counter molecules that are suppressive, organ transplants. There's so many different ways that this could go. So that's why we're excited to work on it, and hopefully we can build these devices and have them function as we would like them to, and demonstrate that they're safe and effective, because I think it will change kind of the course of medicine, and really kind of reduce a lot of the burdens on the patients as well as the cost. Like staying in the hospital is quite expensive, and patients don't like it. So if we can kind of remove some of those
Yeah.
obstacles, that's going to
We
be,
hate it.
yeah, I mean, it's not fun for anyone. So
Well, I appreciate that you guys are approaching this from just a different angle. I feel like so much cancer research right now is in finding targeted therapy. And while I think that's amazing and we do want more targeted therapy, it does feel like cancer is always like one step ahead. Like the moment we find a way, even people who are taking Herceptin at some point, they become non-responsive to it. So I think that cancer is always finding a way to mutate. So I appreciate that you guys were looking at this problem and saying, okay, well, how do we not treat it as a static disease? And also, how do we do local delivery? And how do we solve some of these problems that just pouring billions of dollars into targeted therapy? How do we solve some of the problems that they're going to face anyways? That even if they do develop these drugs, they're going to, maybe face these problems down the line. Feels like you guys are thinking a couple steps ahead, which is really nice.
yeah, I mean, that's the whole model of ARPA-H. Again, it's a new funding agency that was just set up 2 years ago. And it's really kind of, and there has been this model for DARPA. So I don't know if your listeners are familiar, but DARPA is part of the Department of Defense, and it brought to us things like the internet and GPS, and it's these kind of like big investments to think about how do we get some of the smartest people in the room focused on a really difficult problem that could probably a solution maybe emerges on its own within a 20-year horizon, but we need it so urgently that if we threw more money at it and brought the right people together, we could get there in the next 5 years, right? DARPA has been doing this for decades now, and we've all benefited from a lot of the technologies that have come out beyond the military so now with ARPA-H, they're intending to do this for health, for medicine.
Yeah.
A lot of the medicine today is, in the United States, it's like sick care, where it's like,
Yeah.
when patients are really sick. But I think we need new technologies to enable actual healthcare. How do we get people to be healthier, new platforms? That's what's exciting about this endeavor, is if we can, solve the problem of regulating someone's immune system to eliminate cancer, we can apply the same platform to regulating their metabolism to avoid obesity. We can, regulate hormonal pathways to control fertility. It just opens up so many new avenues for healthy living, as opposed to just end of sort of, like it's already late. People already have a lot of disease burden.
Yeah.
How do you remove that?
Yeah. Can I ask you right now, because you're doing it with interleukin 2, would you be able to do it with something that was like a chemotherapy? Because obviously interleukin 2 is something that's made in the body. You guys are doing cell signaling with it. But would you be able to do a drug delivery with like putting cisplatin in there? Like or would that just not work because we wouldn't be able to like signal to the body to start forming cisplatin?
Yeah. No. So yeah, that's challenge. That's kind of like not in the scope for us to do, and also difficult to do, because those type of drugs are all synthetically made and not anything
Right.
that biology itself produces. So we're limited to things that already exist in biology. But a lot of the interest, the reason why I think this is the way to go is because the body can address a lot of its own malfunctions. We have pathways to do it, and there are biological molecules that itself encodes, like IL-2. There's other kind of molecules in the pathway as well, like IL-12, TNF alpha, and countering molecules, right? And so I do think that the riddle is going to be solved by figuring out the right combination of the molecules that the body itself produces, as opposed to having to go to more of these synthetic drugs that, are made in kind of laboratories, but they've never really been.
Complicated ways.
Yeah, and they
Yeah.
have a lot of
So,
off-target effects.
yes. Yeah. So with something like I I want to make sure this isn't like that I'm not asking you a dumb question. Okay, but something like checkpoint inhibitors, like a Keytruda, and people have to get tested to see if they have the right, markers and mutations to see if this drug is effective. Are you guys kind of bypassing that? Like, would IL-2 like not be reliant on certain mutations? Would it be more ubiquitous across the board that we could deliver it to cancer patients?
I think we're going to prioritize cancers. I mean, to start with, we have to pick kind of like one target, and we're starting with ovarian cancer because that's
Right,
one particular disease
right.
where there should be an immune response, but there isn't. And I think,
Mm,
a platform like us could maybe have an impact there. And then we
yeah.
will, broaden it to other types of tumors as well. Initially, it's going to be tested in patients that have not benefited from traditional methods of managing their cancer. But some of those same things are important for us to consider, like whether they have upregulated PD-L1. So we can actually make Keytruda from the device, and that's in the plan. So we're going to be making the checkpoint therapies, not just IL-2. It's a combination of many immunotherapies. That what's cool about this is we
Oh,
have
cool.
one drug product, which is the device. This one drug product can make multiple drug substances, which is like IL-2, Keytruda. So you can think about in one patient the ability for the clinician to you just get one procedure, but then the clinician decides you need X dose of Keytruda, you need X dose of IL-2, and let's try this for a day and then see how your immune system responds. And then, maybe it's working great and you keep on that. But if it's not working, okay, now let's try different immunotherapy. And that's what's cool is not just the ability of changing dosing, but also going through the combinations um, and figuring out what makes sense to for that particular patient.
And that's incredible. And that's coming from these little drug factories that it's you guys are using cell signals to do that. So like it's your body that's it's basically turning on like a light switch and being like put light here. And then it's your body that's making it.
Yeah, it's being made in your body
Wow.
by these engineered cells, and it's all possible because like Keytruda is made by cells in a bioreactor. We're just bringing that manufacturing
Right.
right into the patient, and we can control that cell. Actually, we use electrical signals to turn on production of these cells. But yeah, so that's exactly how it works. It's like make this molecule in this moment in time for the next 8 hours or for the next 24 hours, and then as it's doing it every 10 minutes, it's taking a reading as to what is happening to the immune system. Is this the right combination?
Well, that is just like the coolest, excuse my language, fucking thing I've ever heard because I just think that really is like when I was trying to figure out what can I do to give myself an edge in cancer, I had this 7% survival rate and luckily my oncologist was like, "Hey, you're going to be in that 7%. I believe in you." Which not everybody has, but you just sit there and you feel hopeless. And then the more I learned about cancer, you know, the more I was like, oh, this is a very difficult problem. Like, there's a reason why there's no one cure. It's because it's 200 different diseases. It's mutating all the time. We have risk of recurrence. And then all the treatment that we have to give you can cause secondary cancer and it ruins your immune system. And it's just this snowball effect of what cancer does. And it's just incredible that you guys are putting a device in somebody that can then start making these immunotherapies. It's your own body making it with these signals, electrical signals, and then it can teach your body how to attack the tumor. And then not only that, then if cancer cells come back, which we know they often do, your body is primed to keep attacking those cancer cells. And that's incredible. Thank you
Hmm.
for working on it, I guess is what I wanted to say.
Yeah, no, I mean, we're very passionate about this and working really hard. Hopefully, it's successful. It's a difficult problem, but
Yeah.
we have a big team, and a lot of smart people are working on well, on our team, but also just across the board. I think we're in a really exciting time because of our understanding of genomics as well as more understanding of how the immune system works. I mean, the immune system is the reason, I keep saying this, but because like the more and more I'm super convinced that, okay, it's a failure of the immune system. The solution is to fix that problem. That's why we have cancer. So it really is an immune system problem. We just need to figure out how to activate the immune system the proper way. And this should really, for a vast number of cancers, be impactful.
Yeah. And I mean, it's obviously failure of the immune system is one of the hallmarks of cancer. And if all of our mutated cells that I used to not understand, how can something be pre-cancerous? And it's because you don't understand this sort of like step schedule of mutations that things go through, and then they become, cancerous. They start, they go from benign to cancerous. And so this idea that if our immune system was recognizing those threats from the beginning, we just wouldn't have cancer, So you're exactly right that it is a failure of the immune system, because if our immune system had looked at our cancer cells and killed them to begin with before they became tumors and before they went through angiogenesis and before they, mutated to the point where we can't take care of it anymore, then we could really solve the problem of cancer. Is there anything else that you want to say about it? Did I miss anything that you just want, that you're dying for people to know?
I mean, I think we covered it all. Like, again, we're very appreciative of this support from ARPA-H. It's great that the government is kind of now has like a health version of DARPA that really is focused on these big transformative things. I think that was the missing piece because pharma companies, they're very good at creating like iterational products and really perfecting them, but something like this is too risky for them to take on. And that's what's unique about this opportunity for us is to have the financial support from the government to actually build something that could be transformative.
That's amazing. Well, thank you for your work on it. And one last question. You guys are going to hopefully have clinical trials on this device in the next 5.5 years. Is that the goal?
Yeah. So we are working on getting this into first-in-man trials within the next 4 years. And within the next 5.5 years, we should have data on safety and initial signal of whether this idea is working or not. And then, if things continue going well, then we would do more trials. But yeah, that's the goal, is to get this as quickly as possible in the patients, learn how well it's working, understand the flaws, fix the bugs, and, keep iterating, building upon this until we have perfected it such that it's effective in, helping people eliminate their cancer.
No pressure at all. Just... Well, thank you for being here. And, if you ever want to go solve the problem of small cell neuroendocrine cancer, please. We're so rare. No one's studying us. But thank you for being here. And sorry, I cut you off.
Oh, no. I was just going to say, I think what's cool about this, again, is if we figure it out for one cancer, then I think, it will have broad implications for others as well.
I agree. Well, thanks again. And thanks for chatting with us and explaining all of the science. And I hope that in 4 or 5 years, I'm going to be talking to you and you're going to say, great news. It works perfect.
Sounds good. I hope so too.
Right. Yeah. I hope so too. All right. Well, thank you.
Thanks. All right.
I'm just pushing.